All right, guys, you guys are doing great, and we've already walked through a large number of different cell components. So what I'd like you to do is challenge yourself by looking at this image on Socrative, identifying the type of microscopy. Then I want you to identify this component of the cell right here, and then I'm going to ask you to describe its function. So please take your time, test yourself, see how you're doing, and then come back and I'll share with you my answers. All right, so this is exactly the kind of thing that you're going to be doing on the exam, so let's see how you did. The first thing you needed to do was identify the type of microscopy you've seen. Well, this microscopy is going to be revealing a lot of information, individual kinds of components of the cell. So I can confidently say that this is electron microscopy. Now I need to decide if it's scanning or transmission electron microscopy. And since I'm not seeing a three-dimensional shape, I feel confident that this is transmission electron microscopy. When I have to identify this organelle, I need to look at individual structures. I'm definitely seeing a lot of membranes, so that's a good way for me to be able to say I need to be thinking about the endomembrane system. It's not a pretty round circle, so I know that it's not going to be the nucleus. And I'm not seeing any individual dots, so I don't think that this is going to be the rough ER. But I also have to decide if I think this is the endoplasmic reticulum, the smooth component, or do I think this is the Golgi? Well, since this structure is not continuous with other membranes, but it's unique and isolated on its own, that tells me this needs to be the Golgi. Now, some of you might have also taken a look and identified that we have some mitochondria out here. We have some free ribosomes and floating around in the cytoplasm. Maybe you even recognize that here is actually a piece of endoplasmic reticulum. It's just a rogue cisternae shape that's floating around. Now, when we have to identify the function of this organelle, there's a number of different explanations that you can use. But that explanation needs to give you some context and it needs to be specific. So here's a full credit example of an answer for the exam. The Golgi is the site where glycoproteins and membranes from the ER arrive. Remember, if you're talking about the rough ER, you should be describing glycoproteins, and if you were talking about the smooth ER, that's the site of phospholipid synthesis. But the Golgi is going to be where both of these products are received. In this case, via the cis side, when vesicles formed from the ER merge with the cis face of the Golgi. For further modifications, meaning those components get modified inside of the Golgi, they're sorted and they're sent out to the rest of the cell via the trans side and using those vesicles. It's sort of like an Amazon processing center. All right, here's 4 more questions for you to try. Pause the video and come back when you're done. The first question is a true-false statement. The Golgi apparatus can be found in most prokaryotic cells. Well, this is false. And even though you're used to thinking of the fact that prokaryotes like bacteria have no nucleus, I want to make sure that I remind you that these is still membranes, that Golgi is still composed of membranes. So prokaryotic cells have no internal endomembranes, and that's because they're prokaryotic. So the Golgi is not found in bacteria or other prokaryotic cells. The function of a ribosome is to synthesize proteins. This is a process known as translation. What macromolecule is a ribosome? Hopefully, because it's an enzyme, you knew that it was a protein. But it's also going to be composed of a molecule of protein and RNA. So there's some unique structures here for ribosomes, even though they're enzymes. And which of these attributes belong to the rough endoplasmic reticulum? They're going to have ribosomes that are bound to the surface, and those ribosomes are going to synthesize proteins inside the lumen of the ER. These are specialized proteins where sugar groups get attached, so we call them glycoproteins. Now, the next part of this class, we're going to be talking about the cytoskeleton. It's a network of protein fibers that support the cell both in structure and shape. We're going to identify the structure of these protein fibers, but I also want you to recognize that there are additional functions that each one of these cytoskeletal components actually has. Each one of these functions is going to be a component not only of their location inside of the cell, but actually their nature and their structures. So here's a lovely example of one of these functions, which is due to motor proteins. Motor proteins are proteins that can actually move inside of the cell. So along the bottom of this screen here, you're seeing a microtubule. And then this is a motor protein that's actually walking along the surface of this microtubule. And because it's also attached to a vesicle, like the vesicles that you've seen moving along in the endomembrane system, it's going to be bringing that vesicle with it. So it's motor proteins like this one here that actually walk and move these vesicles through the cell. Now, this microscopic image that I have at the bottom here, would that be light or electron microscopy? We're seeing individual protein fibers and a vesicle inside the cell. That's definitely electron microscopy. 3D shape or something that we're seeing a slice of? Yeah, this is definitely a 3D shape. This is a scanning electron micrograph. ATP is necessary for each one of those steps that you saw that motor protein take. And so this is going to be one way in which we're spending energy in order to provide movement in the cell.
Containing the dark pigment melanin as they travel along microtubules in a fish epidermal cell. This movement of melanin granules enables the fish to change its color to avoid predators. Rhodamine-labeled tubulin was used to visualize the microtubules. Notice how some of the microtubules appear to move as tubulin monomers are added to one end and removed from the other. Credit, Gary Borisi, Northwestern University Medical School.
So microtubules are going to be the thickest cytoskeletal fiber. That's the one that you were able to actually see each individual fiber in that last image. They're constructed of hollow rods, which is how they're able to get as big and as thick as they can. They shape and organize the cell, for sure. All cytoskeletal components do. But these are actually those structures that you saw the motor protein walking along, because those motor proteins can guide the movement of organelles. This includes vesicles, but also larger organelles as well. They also have a function which includes guiding the movement of cilia and flagella in eukaryotic cells. And they do this using the same types of motor proteins that we saw. The difference is that they take two microtubules next to each other, and then the motor proteins walk one way or the other, or one way or the other. So instead of walking entirely along one track, going from one side of the cell to another, they take a step forward and a step back. They can also separate chromosomes during cell division. So the mitotic spindle, which some of you may remember from high school, that's the thing that those chromosomes are attached to when they actually divide during cell division, is composed of these microtubules. Each microtubule is organized around a central centrosome. It's a pair of what are known as centrioles. This centrosome is an organizing center, but it's also something that you can see if you know to look for it. And it helps you to identify this component of the cytoskeleton. Each centrosome is a pair of centrioles, so you may see this structure at 90 degrees of one another, and so there's also a characteristic shape here. When we take a look at how these microtubules are organized, not only are they a network throughout the cell organized as a centriole, but when they form structures like cilia and flagella, they're going to be creating these unique kinds of tubular arrangements with a central pair and then pairs arranged around that central pair. And the motor proteins responsible for the movement of these organelles are connecting that central pair with the ones around it. We call these motor proteins dyneins, and they're connecting those doublets together. Now recall that this is a eukaryotic structure, so this is how eukaryotic flagella move only. I just want to point that out because it's a little different from bacteria. Flagella are single or very few numbers of very long protrusions that are especially important for cell movement. Cilia are shorter and more numerous, and while they can allow movement, such as in paramecium, they can also have other functions, such as the cilia of your intestinal cells, which increase surface area and allow you to absorb nutrients. If we take a look at these motor proteins like dynein walking along microtubules, dynein is going to be a very specific motor protein that when it walks its feet along the microtubules can walk in such a way that it creates the bending of cilia and flagella.
Observe the sliding of microtubule doublets that is responsible for the movement of a sperm cell flagellum. Tiny gold beads, 40 nanometers diameter, were attached to opposite sides of this flagellum, and ATP was added to stimulate movement. Notice how the beads move relative to each other, demonstrating the microtubule sliding action that causes the flagellum to bend.
Now, microfilaments are going to be different. Microfilaments do not have a central organizing component. When we talk about how microfilaments support cell shape, we're going to be looking at these very thin fibers. They're actually a twisted chain of actin monomers that form a structure known as a cortex. It's this ring around the surface or the outside of the cell. Cortex means crown, and if you take a look at this image right here in red, you'll see those microfilaments forming the cortex and how it looks almost like a crown-like shape. These can also form microvilli, which is another type of protrusion in the cell, but these microvilli are not going to be capable of movement, and they're not going to be composed of microtubules. But microfilaments can participate in cellular movement, not as cilia or flagella, but as pseudopodia or muscle contractions. Here's an example of microvilli. You can look at the two images here on the right and try to identify which one of these would be an example of a structure that contains microfilaments and which one would be a structure that contains microtubules. Yeah, so this structure here on the top, this would be an example of microvilli, which contains these microfilaments. As where if you compare the bottom here, we have that distinct doublet, central doublet pair of microtubules. These are much larger in diameter. You can even see the hollow rod-like shape, much larger, and then that ring of doublets around it. This is an example of cilia. Now these actin filaments, remember actin is the monomer that forms these microfilaments, also interact with a protein which includes myosin. Myosin is responsible for muscle contractions. This motor protein doesn't just have 2 feet, it has many feet. And so when you get a contraction, it walks fibers together, and then when that relaxes, it walks them apart. So myosin is still going to be a motor protein, but its structure is going to give it a slightly different function. Pseudopodia, which is, for example, the amoeba that you see here on the bottom, they move the cell, but they do so in a unique way. They don't look like cilia or flagella. They actually have extensions, movement, these protrusions, where the cortex of the cell expands by polymerizing new microfilamental fibers, adding actin, and extending the boundary of those cells.
See the actin microfilament network in the lamellipodium of a Xenopus keratocyte that was cultured on a glass cover slip. The video shows the cell in motion, followed by electron microscopy to visualize the extensive actin network present in the lamellipodium. Notice the extensive branching of the actin network highlighted in yellow. Credit, Gary Barisi, Northwestern University Medical Center.
So you can actually see these little twisting doublets, which are the cortex of the cell. And it's important to notice that that cortex is found on the surface of that cell because that's what allows it to create this pseudopodia-like movement. Intermediate filaments are larger than microfilaments but smaller than microtubules. There's only one example of intermediate filaments that I want you to know, and that's the nuclear lamina. Intermediate filaments are going to be different. They're more permanent cytoskeletal fixtures than either microtubules or microfilaments. Microtubules and microfilaments can be created or destroyed. They can polymerize or depolymerize to provide that movement, that network that adapts with the cell. But these intermediate filaments are more permanent structures. That example being the nuclear lamina, which you can imagine is an important structure to have. So here's my last question for you, because I'd like you to remember it's important to connect what we're learning now with what we've already learned. So tell me, which of these arrows indicates the nuclear lamina? Pause here if you need more time. Good. Here's our nuclear lamina, that protein network which is found underneath the surface of the nuclear envelope and providing additional shape and support to that nucleus.